my name is David. I'm one of the science teachers here at Hamilton Education. Today I'm going to be going over the science test for the ACT 495. All right, if you're ready, let's begin. Question one. Question one is an interesting passage where it has a lot of weird things in there that most students don't understand. But again, you can go to the questions first and help figure out the values. So definitely, let's look at the first question. Based on table 1, which of the following graphs best compares the viscosities of 1% aqueous mixtures of gums W, X, Y, Z at 45 degrees Celsius and a resting time and temperature? Well, looking at that, we have table 1, which is right here. And then we are looking at viscosities, which it says viscosity here. And we want 1%. And we want gums WXYZ at 45 and resting time at 75. So we have 75 right here, and the 45s are here. So which one was the greatest? Well, we have 6,000, which is the biggest. So W should be the first one, or the tallest. So B looks pretty good. So does C. And then look at the rest of it. So is X very small, or X is the medium? X is the medium one here, which is why that the final answer is B. And so that's kind of one thing that's weird. Like, I didn't look at the rest of the stuff, but I used the table to figure out this information out. And we'll do this pretty frequently. All right, let's do the next one. Question number 2. Based on the table, if just prepared 1% aqueous mixture of gum Y is allowed to sit for 100 minutes at 65, the viscosity would most likely be what? So we want to go to gum Y, and we want to go to 65. And one thing I noticed is that 100 minutes is between 800 and 1,000. So we want to pick the answer as 800 and 1,000. C is the correct answer. Question number 3. Consider the viscosity shown in figure 1 for a gum concentration of 2%. So we're going not to the table, but the figure. What is the gum from the viscosity from lowest to highest? So looking at the 2%, 2% is right here. Gum Z is the smallest, so we're crossing off A and B. And Y is the highest. So Z to Y, that it would be C. C is the correct answer. So again, very quick. Question number 4. Under which conditions that are the basis for figure 1, a 1.3% aqueous mixture has the highest viscosity? So we want 1.3, which is not right down the middle, but a little bit over to the side. And looking at the highest, so earlier we said gum Y was the highest, but at 1.3, W is beating out Y. So that means that the answer would be A. Question number 5. Based on table 1, a 1% aqueous mixture of gum Z at 30 degrees Celsius at a resting time of 75 minutes would be what? So going back to table 1, gum Z and at 30 degrees Celsius, so that's between the 25 and 45, at 75 minutes, so it's between 800 and 1500. So that answer is B. All right, I believe this is the last one for the first passage. Under the conditions that are the basis for figure 1, an aqueous mixture of gum viscosity of 100,000 CP at a lower concentration than any other 3 gums. So figure 1, 100,000 is right here. And we want the lowest concentration than the other 3 gums. So who hits it first? Well, gum Y hits it first at a percent of 2. And so that's why C is the correct answer. All right. I hope we found the first passage as quick as I did. Let's go on to the next passage. All right. In the next passage, we have to deal with something called biodiesel and some chemistry knowledge of converting things with a catalyst. But once again, it may be helpful to check out the questions first. So looking at question number 7, if the student had tested 60% BD by volume in study 2, A would have been at 1746, would most likely have been blank. So we're looking at study 2. So you might have to scroll down to study 2. And there it has the A is 1746, and we're doing 60% BD, which I see is the x-axis. And so the value for the A is about 0.6, which is between between 0.55 and 0.65. So the answer is C. All right, question number 8. In a study 2, the among samples tested as the percent volume BD increased, the A did what? Well, looking back at that graph, as BD went up, A went up. So it increased only. All right, next on the list. Based on results of study 2, which fuel sample in study 3 most likely had the sample blank? So that means we're comparing 2 and 3. So most likely had the smallest A. So looking back here, I noticed the smallest A compared to the percent BD was the smallest. So looking at these, the smallest BD was W, and that's why the answer was A. All right, a little bit of chemistry background knowledge. The production of BD is describing the passage best represented by which of the following chemical reactions. So on the left side of the arrow is reactants, and the right side are the products. So BD is a renewable alternative to traditional petroleum diesel, and it's prepared by reacting soybean oil with methanol in the presence of a catalyst. So we want soybean oil and methanol on the left. And the only one that has that is C. And then we don't make catalysts, we make the FAMEs, whatever that is. But yeah, so the answer is C. Question number 11. Suppose that study 1, the student had measured the absorbance of wave number 600 through only 1600 instead of through 1800. So looking at this graph right here, we're cutting it off at 1600. And based on figure 1, would students most likely have measured an absorbance in study 2 at a wave number of 11752 or at wave number 1464? So what do we want to do for that one? Well, looking at this, right, we're looking for the highest absorbance. And for not for PD, but for BD. And so you may be looking at this point right here. But then right here for answer A, that doesn't work out because of the fact that it says because PD, not BD absorbs more, that would be the dashed line. So it's not A. For B, the wave number 1172, because PD, not BD absorbs strongly. That sounds pretty good. How about the other two? Well, at these other two, it doesn't absorb as strongly. So because it doesn't absorb as strongly, we're going to want to pick B. Also, the passage mentioned it too, that they wanted to separate it. So one of the reasons why they did the 1700 before was because BD was pretty high and BD was pretty low. If you kept it the same, then it would be hard to separate the two values from each other. All right, so a little bit trickier, but let's go on to the next question. Consider the sample that contains only FAMEs. All right, that's interesting. So because normally that's the product after, so that would mean that there's no BD in there. Based on results of study 1, with the sample strongly absorbed wavelength of 900 or 1250. Well, if it has only FAMEs, then it has no BD. So how can we figure this out? Well, it says BD contains the FAMEs, and BD absorbs more strongly at 1200 than it did at 900. I'm sorry, Reed, I have to redo that question. Ooh, I went to the answer. Didn't explain it. All right. On a 1, on a 2, on a 3. Question number 12. Consider a sample that contains only FAME. So back in the beginning, when we did the chemical reaction, we had the BD is prepared by reacting soybean oil and methanol in the presence of catalysts forming compounds called FAMEs. So that's how BD is produced. One thing it mentioned is PD has no FAMEs. So if a sample only contains FAMEs, then it's only going to be BD. Based on the results of study 1, would the sample more strongly absorb light at 900 or 1250? So 900 is about here, and they're both pretty low. But 1250, BD is higher than PD, so it looks like 1250 is a better answer. So we're going to cross off A because this is 900. We're going to cross off B because it says 900. Now C and D are both better. So PD contains FAMEs. No, that's incorrect. BD contains the FAMEs. And also it says BD absorbs more strongly at 1250. That's also not quite right. They're about equal. So that's another way of figuring out D, but we can also confirm it by picking it. At 1250, it's higher than at 900, and BD contains FAMEs, and this is the best answer to the question. All right, this is the last one for passage 2. Question number 13. Consider the percent BD listed in table 1 for sample Y. A 10-liter volume of sample Y would contain approximately what volume? Well, this is pretty easy. We go to table 1, and Y had about 4.8%, or basically 5%. 5% of 10 liters is not 5 liters. That would be half a liter, or 50%. So we don't want 5 liters, but we want 0.5 liters. And in order to convert liters into milliliters, you times it by 1,000. So that's how you do it. On to the next. All right, passage 3, question 14. According to figure 1, which two regions are most similar in thickness? So we want to look at figure 1, and we're looking at the thickness of it. So how can we determine that? Well, inner core and the outer core. So inner core and the outer core, we have these two, where they have them labeled right there in the figure 1. We have the distance. We're not looking for distance. And we have gravity here. We're not looking for gravity, but we probably want kind of the, not density. Density would be kind of related to like the mass or volume or how dense something is. So I think what we want to do is we want to look at the radius and see how much the width of these blocks are. So the first two are reasonable. It's about half the thickness. Let's see if there's a better one. The inner core and the upper mantle. Inner and upper mantle, those are much better aligned. Outer core and the lower mantle. Oh, they're both exactly the same. So although you might have been tempted to pick B, C is a slightly better answer. So, and then lastly, outer core and the upper, outer and the upper. Yeah, those are backed off. So this is a pretty tricky question. Again, you could have picked B being a little premature, but this is a little bit thinner than that. And then these two are about the same. So on to the next question. Question 15. According to figure 2, the innermost 30% of the Earth's mass is located at R between 0 and where? So we want 30% of the mass. So going to here, and that would be an R from 0 to about 3,500. Maybe, yeah, 3,400. Looks pretty good. So the answer was A. Figures 1 and 2 indicate the Earth's radius is approximately what? Well, figure so it kind of, you maybe draw a circle, but here's the Earth in the center, and then this is the radius. But we also have this air all around it. So we don't want to include the atmosphere. So the Earth radius should be from 0 to about 6,500, 6,400. And you can kind of see 100% of the mass of the Earth is about 6,500. So either graph could help you get the answer D. All right, question number 17. Based on figure 1, the approximate of the Earth's gravitational field at 14,000. So if you've been taking physics 1 or physics C mechanics, you might be like, oh, it's 9.8. I didn't know that, blah, blah, blah. But at 14,000, it's further from the Earth, so the gravity could be less. And more importantly, you want to look at this graph, the gravity curve. And we have it at 14,000, so it's a little bit beyond here. So I'm going to say between 2 and 0. So less than 0.5, I think that's too much of a difference. And 0.5 to 1.5, I mean, maybe it's a little bit there, but I think it's between 1.5 and 2. So I think C is the best answer, but definitely it is close. And again, you can use your fingers and kind of like line it up and try to get a better approximation. All right. Question number 18. On average, Earth's crust is about 30 kilometers thick. Based on figure 2, the crust approximates for what percent of Earth? So this is a pretty tricky question. Excuse me. Because a lot of people want to start with a 0 to 30 here. But what you want to do instead is we want to go from 30 kilometers from the edge, because the outer crust is on the outer edge. So we want to go from here to there approximately. All right, so what seems like the best answer? It still kind of looks like about 0.1. Because these are going to be in 200 kilometers. So you wanted to look at that point right there to kind of help figure it out. But yeah, it's not that much of a mess. What about 10%? Well, if you go from 110, that's 90%. And that would not be 30 kilometers, but that would be approximately 300 kilometers. So that's another reason why it's not B. Last question for passage 3. So consider 2 hypothetical 1 kilogram rocks are located at 2,000 and 4,000. Which of these rocks would either weigh less? Now, again, if you're in AP Physics 1 or AP Physics C, this might get you mistaken because you might remember your basic understanding we're like, oh, if two rocks have the same mass or the same weight. But that changes as you go away from the Earth. Like if you're on the moon, the weight drops because there's less gravity. And if you go inside, the gravity can change as well. So what we want to do is instead use the graphic to figure it out. So at 2,000, the gravity is about 6, and at 4,000, the gravity is higher. So because the gravity is higher at 4,000, the weight will be higher at 4,000. So which one weighs more or weighs less? So we want the 2,000, so A looks pretty good because the gravity is less at that location. So that seems pretty solid. How about the other answers? 4,000, no. Neither rock, they have identical masses. So that's definitely taking your outside knowledge question and using it against you. Neither rock's value of gravity is for the same, and they don't have the same gravity. The curve changed. So definitely, it's a kind of tricky one, but hopefully that makes more sense on why the answer was A instead of the other three. Okay, so we're on to the comparing viewpoints. So what are we doing? Well, generally speaking, it's pretty helpful to write down notes for these types of questions. So let's see what notes we can write down. And then I basically keep them off to the side so we can reference back to them. So first thing we want to do is talk about the question. So biological aging is the process in which the function of animal cells gradually decline and cause cells to age. For students, there we go. And then it can propose the explanation for why that occurs. All right, so we're looking for reasoning why we age, basically. Let's make some notes down. Student 1, what does he say? Or they say? BA, biological aging, is caused by ROS, the reactive oxygen species produced by cellular respiration, by cell respiration. ROS damages proteins and lipids. The cell produces, makes antioxidants, but the antioxidants is less than the damage, it looks like, and therefore the ROS accumulates over time. All right. How does that compare to scientist 2? Scientist 2. Biological age is caused solely by the formation of cross-links, so not ROS. It does have the word protein there, so bonds between proteins. And they form clumps. The clumps give you cell, interfere with cell functions. Clumps make cell age. And then although RS damaged protein, so RS does exist, the damage never occurs because cells producing cells is always greater than. So that's a counter. The antioxidants is more than the damage. So that would probably be a question that they will tend to ask about that. Let's look at scientist 3. Biological is caused solely by DNA damage. By environment. So like maybe sunlight or smoking. The extensive damage is caused by seeds' ability to repair the damage. So it doesn't say anything about ROS or damaging like that. But it does say about cells' ability to repair, which is similar to like the antioxidants. Again, ROS does occur. It does not accumulate in the cell. Does not accumulate in cell. And then it does say cross-link does occur. Do form clumps, but it does not affect cell function. So again, mentioning the same thing that scientists 1 and 2 did. All right, and then lastly, scientist 4. Biological is caused by lipofusion. BA is lipofusion, which is a oxidized lipidide. Sure. Lipid by cell respiration. Well, they do repeat the word cell respiration as the other scientists. It forms clumps. So also forms clumps, different types of clumps that interfere with cell function. They do produce ROS, so ROS does exist, but it never accumulates. So that's like scientist 3, does not accumulate. And the cross-link is short-lived. The cross-link clumps don't exist. Cross-link clumps don't exist. And the damage just in the cell does never exceed its ability to repair. And then so DNA damage does not cell repair. All right, now that we have a big overview of notes on our paper, we can kind of compare them. So let's look at the first question. Which instance, if any, claim that biology is caused by a substance produced by cellular respiration? Well, looking at this, cellular respiration was definitely student 4. And then we also had cellular respiration from student 1. So 1 and 4 sound pretty good. Did 2 and 3 mention it? Well, 3 definitely didn't mention it. And also, we have to have 1 and 4, so we can't pick C. Suppose we determine that the rate of biology in an animal cell directly proportional to number of chemical bonds formed between proteins. So again, protein forming was between scientist 2. The finding would be most consistent with which one? Student 2. Based on student 2 explanation, the substance present in cells that are most directly involved by the aging. Well, student 2 is all about proteins. And so this is an outside knowledge question. You have to recall that amino acids make up proteins. Fatty acids make fat, monosaccharides make sugar, nucleotides make DNA. Farnesene is a substance that prevents protein cross-linking. So that definitely would stop student's 2 argument. Which student most likely would predict that the average concentration of carnosine cells of young adults would be greater than the average concentration in the elderly adults? So what could argue is that in the beginning, you have the carnosine which prevents cross-linking, but over time, as the carnosine degrades, you can have more cross-linking occur. So this would tie back into student 2. Which of the students claim that biological aging occurs because a substance accumulates into clumps that affect the cell function? So we don't know about 1 because it didn't say clumps at all. So it's not 1. So that means it has to be 2 and 4. And we did write clumps make cell age and the clumps interfere with cell function. Student 1's explanation would most strongly be supported in which of the following observations were made. So again, student 1, it's all about the ROS and forming, damaging the proteins and lipids and talking about the antioxidants. So increasing the protein links. We don't want protein links. That was not relevant to scientist 1's argument. That was relevant to scientist 2's. Increase the antioxidant concentration. So does that increase the rate of cells aging? No, antioxidants were undoing the damage. There was not enough antioxidants. So probably D. If we decrease the antioxidant in cells, it would increase the rate at which the cells age. That would exactly tie in with our argument. Question 26. Compound X is a chemical that causes genetic mutation. Genetic mutation sounds like DNA damage. Suppose it was determined that the human neurons grow in the presence of compound X age at the same rate as humans grow in the absence of compound X. This would weaken the explanation of which of the following. Well, compound X causes DNA damage, so that would weaken scientist 3, or student 3. What about 1 and 3? Well, 1 was about antioxidants. It didn't say anything about DNA damage or human neurons, so we can't conclude anything about student 1 and 3. All right. So let's see what the next one would be. All right. According to the results of the studies, from which of the 5 leachates was the greatest percent of DOC adsorbed by CM1, CM2, CM3, respectively? So this one has a lot going on. So first off, adsorb is not absorb. Adsorb is to adhere to a substance. All right. And then leachates was definitely something where it involves leaching from things. And the leachate is the resulting liquid that has the compound in it. So definitely, if you come across words you don't recognize, you may want to do a quick scan to find those words and read their definitions. So which of the chromosomes was the greatest percent of DOC for CM1? And we have to look at all the studies. So let's see, looking at here, pine is definitely the highest for CM1. So pine, okay, so it's either C or D. Magnolia was the highest for CM2, and magnolia was the highest for CM3. So D is the correct answer. Question number 28. Question number 28. According to the results of study 3, the percent of leachate DSU adsorbed by CM3, the average across the 5 types of leaves, is closest to which of the following. So we're going to look at study 3, and we have the percent leachate absorbed by CM3, and we want to average across all the numbers. So you kind of want to like see that this is a little high, it's a little bit low. So I'm looking at it and it says like 40 seems like a pretty good number. 30 is too small, and the rest would then be even further smaller. Like 40, 45, it'd be kind of hard to figure out which one to be. So they made it kind of obvious of which one it has to be. And the same as CM2 absorbed a greater percent of DOC in the maple than did CM3, supported by the results of study 2 and 3. So comparing study 2, we want to look at maple. So in study 2, maple had about 5-7%, and in study 3, it had a much greater amount. So is it greater than 2 than 3? The answer is no. So we want to focus on the nose. CM2 absorbs 7% and DOC, where CM2 absorbs 35. That sounds perfectly right. And then this one says 17, which is maybe you're looking at a wrong number or something, misread the graph. All right, on to question 30. Question number 30. What was the independent variable in each of the 3 studies, and what was the independent variable across all the studies? So it kind of looks like, based on what I see, is that the independent variable in each of them is kind of like what type of tree it would. And then we have a lot of leachate in here, which was the lake water. So let's see which one best answers that. We have lake water, which sounds pretty good, and type of clay materials. It kind of seems right. Let's see what B is better. The type of leaf leachate. Okay, so I know it said that leachate was a liquid result by it, and that kind of really seems related to the water, but the graph says leachate on there. So although lake water sounds reasonable, I think that the better answer here would be B. But yeah, the clay minerals looks pretty good. Let's see if there's a different answer. Mass, no. Mass leaves, no. So then the best answer was B here. But A is very close. So question 31. According to the results of studies, which of the 3 clay minerals, if any, reduced DOC in the oak leachate by more than 50%? All right, so we're looking at all of the studies again. So, and we're looking at oak specifically. So oak has 30%, which is not more than 50%, so not CM1. Oak has small amounts in CM2, so not A, B, or C. And again, it says more than 50%. So if you're tempted by CM3, then that would be right, probably on it or below it, but it's not more than. So that's why the answer was D. Is a mixture of any one of the leachates and any one of the clay minerals properly considered a solution? So you can definitely read over back what a leachate is. So going back to here, green leaves were collected from 5 types of trees around the lake. 5-liter volume of the lake water is filtered, remove all solids. The following procedures were performed. 100 grams of the samples were mixed with a liter, and the mixture was then placed in dark for 10 weeks, 4 degrees Celsius, while leaching occurred. So the water is extracting things from the leaves. At 10 weeks, the mixture was filtered to remove all solid particles. So then basically the remaining amount is called the leachate. And then they did the leachate mixture with a CM. But the thing about it is a solution is going to be properly blended. So like imagine like Gatorade is a solution of like water and electrolytes and sugar. So that is something you don't have different particles floating in there. So this is more of like a mixture. So did the clay minerals particles dissolve? No, they did not dissolve. If they did dissolve, then it would be considered a solution. No, because the clay minerals are dissolved, not dissolved, not dissolved. So definitely kind of tricky. It's an outside knowledge question from your chemistry class. I think this is the last one. We might have 2 more to go. This is the last one. Perfect. All right, last one for the passage 5. In the lake water, DOC is broken down into simpler compounds by electromagnetic energy, also known as light. What action was taken in the study to prevent this process from occurring? So that's called experimental design, where you want to go back and read over what they did to prevent it. And it said earlier that they placed it in the dark. So if it's placed in the dark, light can't reach it. Was placed in the dark, was placed in the dark. So those are all pretty good. each mixture of leaves and filtered water was placed in the dark. Each mixture of the filtrating claim was placed in the dark. So that's study one, where it seems like it's here, but it was earlier, back when they had the leaves and the water that they placed in the dark. So the answer was A. All right. Question number 34. Which of the following figures best shows the orientation of PD5 before it was rotated 90 degrees? So PD5 is scrolling down a little bit further. Yeah, so right here, we're looking in the current 2, it had placed it in there. And then it mentions that they were turned 90 degrees. So what did it look like before it was turned 90 degrees? And so the answer, as you might guess, was C, because a PD5 was the same as, I think, PD1, and then they rotated it 90 degrees to make it look like that. This one would be rotating 180. That's exactly how it was. So, and let's go on to the next question. 35. PD8 contains the same type of seeds and was subject to the same growth conditions before being turned as which Petri dish in experiment 1. It was the mutant seeds placed in PD8. So going back to here, the mutant seeds were placed in PD3 and PD4. And then PD3 was placed in the dark for 70. And whereas PD7 was also put in the dark. So the answer should have been PD4. So definitely reading about the experimental design is how we can answer that question quickly. Based on the results of experiment 1, in the absence of light, the radicals of the seedlings have the same response to gravity as the radicals of the WT seedlings. So there's a lot of stuff you might not know. All right, so the radicals, that is something that refers to earlier where we have it italicized, and it says the roots. There's apicotyls, and then, okay, right, the roots are there. All right, and then we're looking at experiment 1, and did the absence of light affect it versus the WT? So absence of light was PD1 and 3, so these two. And then we have the wild types right here. So we have wild type here, mutant here, and then dark on the left, light on the right. So which one do we want to pick? So in order to compare them, we want to compare the M's to the WTs. So we're basically comparing 1 to 3. And it does kind of seem like they're different. Let's see what the variation. And the orientation of radicals was greater than that. So I kind of feel like it does not have this. I mean, these two have the same, and these two are kind of the same, but definitely there's a lot weird stuff going down there. So I kind of say it's not the same response, so it's no. And the variation in PD4, well, PD4 is in light, so we can't pick that one. And that's comparing the light ones. So then this, okay, so yeah, it's either yes or no. And yeah, you either thought it was the same, so C, or no, they're different. I think that these are growing a little bit normally, but then the mutants grew a lot weirder on the bottom. Question 37. During the 25-hour period in experiment 2, the WTCs were exposed to light from below. Did the hypocotyls more exhibit positive phototropism or negative phototropism? So again, phototropism is something that responds to light. Positive means it goes towards it. Negative means it goes away from it. So looking at experiment 2 and the results of it here, we have the light being grown from below, and that's the radicals. And let's go through it. Reading over it with y'all. We have the radical curvature, but looking at this right here, I don't quite see the answer. But normally hypercross will grow towards the light. So I feel like P5 and P7. So those are the white circles and the square. Okay. Oh yeah. So they have where they're exposed to light from above, and we want the wild type for PD5. So we want PD5 itself. And then looking at that, the radicals grew downward like normal, but I would imagine the hypercodals probably grew the positive. Like it might be an outside knowledge question. So acetylcholine is going to go positive because acetylcholine should grow towards light. Yeah, looking at the key, definitely it was an outside knowledge question where basically you have to make a guess based on the figures. Based on the results of figure 1 for PD2, Arabas is a monoclide or dicot. This is a little tricky, but the word mono means a single and di means two. And then the cot is referring to the cotyledon or the hypocotyledons. And so look at the pictures for PD2. You can see there's two little leaves on there, which is why it was a dicot for two cotyledons. Question 39. Question 39. In experiment 2, each of the features had how many orientations? So basically we had orientations here where basically we had it normal and then we rotated it 90 degrees. So the answer was 2. To evaluate the effect of light on the growth of WT Arabidopsis seedlings, the results of which two features in the experiment should be compared. So comparing them, we would want to compare PD1 and PD2 because in PD1 and PD2, those are both wild types, but one was grown in the dark and the other one was grown in the light. PD1 was in the dark and PD2 was in the light. Sounds good? All right, and let's see how we did. So, yep, thank you all for your time, and I hope you found that helpful. And we even beat the clock. All right, bye-bye.